So welcome everybody. So today, this session, there are three very interesting talks there. So first talk is by Professor Simone Pigolotti, and he's going to talk about dynamics of bacterial replisomes and stochastic replisomes. Please go ahead, Simone.
Thank you very much. So first of all, I would like to really thank the organizer for the invitation. I was saying that I didn't have the chance to attend many talks at the right time because that's not a convenient time for me, but I'm watching all the talks on YouTube. I mean, all the talks that I couldn't watch live, I've been watching them on YouTube, and it's been really an amazing workshop. I wish that next time we'll be able to meet all in person. I have just a slide to tell you a bit about my research interests. So my background is in non-equilibrium statistical physics and stochastic thermodynamics, but over the year I've been somehow applying ideas of non-equilibrium statistical physics to mostly biological problems, in particular in problems in population dynamics, and I've been also interested in understanding information processing in cells. So these two things have been somehow parallel interests in my unit and in general in my research. So today I decided to tell you about one problem which falls within the scope of information processing in cells. But I hope I'll convince you that this is not only an interesting problem for biology, but the kind of tools that one can use to understand these systems are the tools that are typical in stochastic thermodynamics. So let me make an introduction about using non-equilibrium statistical physics to understand replication of information in cells in particular. So stochastic thermodynamics has a long history in describing mesoscopic machines that transform a form of energy into another form of energy. And the paradigmatic example are molecular motors. So this is an illustration of kinesin. And this is because these are mesoscopic systems that operate with energies on the order of the thermal energy. And the kind of question one would like to answer is what are the constraints between properties of these motors that one is interested in? For example, the work they are able to perform, their speed, and how dissipative they are. So the idea is that theories like stochastic thermodynamics can somehow in a general way permit to understand the trade-offs between these quantities. And what I find particularly exciting is to generalize this sort of thinking to molecular machines that process information. So this is the example of a ribosome, the macromolecules that produce proteins. And I see this as somehow a generalization of a molecular motor in which there is an additional dimension, which is accuracy. So how well these macromolecules process information. So this is a chance for us to use these kind of ideas, but think of machines that perform in a way more sophisticated tasks. And I've been working over the years in developing general models in which essentially you have a heteropolymer, and you imagine your machine as something that goes through this heteropolymer and sequentially tries to copy it with a certain accuracy. And I use the stochastic thermodynamics to derive general bounds between these observables, these wished properties that this machine have. So ideally, one wants this protein to proceed with a high speed, low dissipation, and high accuracy, but of course this is not always possible, and there must be thermodynamic constraints between these quantities that one can derive using tools of stochastic thermodynamics. So normally when we talk about accuracy, in this case, would be just how accurate is the copy, how infrequent are errors. But in my talk today, I will talk about a slightly different machine and a different kind of accuracy. So the machine I'll talk about is the replisome. So replisomes is a very large macromolecule. There are a large number of subunits, and this macromolecule is responsible of replicating DNA. So replication of DNA is possibly the most fundamental task that cells need to perform. So all cells, before they divide, they need to duplicate their genome in order to pass genetic information to their daughter cells. And this molecule is very large and it's pretty astonishing. So I like this old review that to give us an idea, that explains what would happen if the DNA was 1 meter wide. If the DNA was 1 meter wide, then the replisome would be as large as a truck, and it will proceed over on the DNA at a speed of 400 kilometers an hour. So this is a pretty astonishing machine if you think about it in these terms. So in particular, what I will talk about is DNA replication in bacteria, because as a theoretical physicist, I want to study a system where things are relatively simple. And before I introduce my model and our results, I need to tell you a few basic facts about bacterial DNA replication. So I was mentioning that this is a relatively simple process, and the reason is that in bacteria, usually the DNA is circular, and the replication always starts at a single well-defined point, which is called the origin of replication, which is this yellow dot in my slide. So here the red represents the template and the blue is the copy. So what happens is that there are two replisomes that attach to the origin and copy DNA in opposite direction. So each replisome copies both strands, because DNA is a double-strand polymer. And so this is the situation. So at some point in time, these two replisomes start from the same point, go in opposite direction, and each one has copied. So this replisome will have copied this stretch of DNA. This other replisome will have copied this stretch. And then they will meet at the opposite point. And at that point, they will together have copied the entire DNA. And then the process is finished. So that's the, in a nutshell, the dynamics of bacterial replication. So what we were curious about is to understand how does this actually works, what is the speed of this thing, and of course we can ask a lot of questions about how is this process regulated as a function of temperature and so on. But for example, an interesting question is how precise is this process? So if this replisome would start at exactly the same point and proceed at exactly the same speed, they would exactly copy half of the genome each. But we know that being molecules, these machines are subject to stochastic fluctuations, and therefore their meeting point must be characterized by some degree of randomness. And the question we have is how large is this randomness? So how precisely do they meet? And this has implications because there can be a higher mutation rates in the place where they meet and so on. So this has also implications for biology. So yeah, these are the questions. I already mentioned them. How precise is this dynamics? How this is regulated as a function of temperature, concentration of nutrients, and so on. There are also some questions that I will not address in this talk. These are some of our future questions, which is what happens if you have mutants in which some of these proteins are missing or are malfunctioning. And I will argue that this is also something that we can study using our model.
Simone?
Yes.
So both of the strands are being copied simultaneously or one by one?
Everything that you see in this figure is a double strand.
Okay.
So that's maybe one way. So each one is copying, it's open, so there is a bubble, but each one is copying both sides.
So both strands are copied simultaneously?
Yes, they are copied in a different way, but that's somehow a macroscopic detail that I won't enter into. So I think what's, for the sake of this talk, let's say that everything that you see here is a double strand. So each replison is copying two strands.
Okay.
This is an important point. Thank you. Thank you for the question. And please interrupt me if you have any other doubts, because again, I have to introduce a little bit of biology, and I want everybody, I want us all to be on the same page. So I'm happy if there is some question in my talk.
I guess you are going to introduce the dynamics, I guess, right?
Yeah, I will. I will. There will be a question at some point. Yeah. So let me first anticipate what's our idea. So people have been characterizing this process in a qualitative way by doing the following experiment. So you can have a growing exponentially growing population of bacteria, you can take all the genetic material that is in there and sequence it and put it in a deep sequencer. And the reason why this is interesting, because then you can look at the abundance of genetic material as a function of the position on the genome. So this is one of these experiments in this paper in PNAS in 2014. And you can see that there is much more genetic material close to the origin than far from the origin of replication. And this makes sense because if the population is growing exponentially, you will have a lot of cells that are replicating their genome. And so since they start always from the same place, there will be a lot of incomplete genomes that have only the origin of replication, right? And it will be less and less likely that these incomplete genomes will have traveled further. So somehow this distribution tells us something about the spectrum of genome sizes that we have in our population. And our idea is to use this information to infer what the replisomes are actually doing. Again, this is just to plant the idea, but I will describe this in a very quantitative way in the following slides. So first, I need to introduce another small piece of biology. By the way, this is this work is under review. Here you can find it on bioRxiv. So there is another tricky aspect of bacterial DNA replication. So this process in, let's say, at 37 degrees in nutrient-rich condition takes about 40 minutes. This is the time the replisome take to copy the entire genome. And this is not fast enough because in nutrient-rich conditions, the cell cycle can be 25 minutes for E. coli. So there is this paradox that the cell division time is shorter than the time it takes to copy the genome. So the way bacteria solve this problem is that in slow condition, things are rather simple. So there is an initial stage in which only the template is present, then a second stage in which replication occurs. Then there is a stage in which the replication has finished and you have two complete genomes inside cells, and finally cell division. But in nutrient-rich conditions, so in fast growth conditions, cells can copy the DNA in parallel. So you can essentially start this process before the previous replication has finished. So in other words, each cell might be replicating the genome for its daughters, but also for its grandchildren, starting replication for its grandchildren, because this process takes more than a generation time to complete. So this is also something that you can observe at the microscope. So you have this really parallel replication occurring at the same time. But this is, of course, is a complication for modeling, because if I have a fast growing population and if I take a snapshot, what I expect is that cells will be at different stages. They all will contain one template, which is the original genome, but they might have multiple incomplete genomes that are the blue ones. They might have these post-replication genomes and so on. So this will be a pretty heterogeneous process. So in order to describe it in a simple way, what we will do is to say, let's forget about the cells. Let's only look about the genomes and say that there are three different kinds of genomes here. The templates that are one per cell. So this is the original genome that each cell has. The synthesizing genomes, so the ones that are being copied at that particular moment, and post-replication genomes. So these are the ones that are finished and they will need to be passed to next generation. And there will be rates connecting these things. So templates can produce start replication, so create a new synthesizing genomes. Replication can finish, so the synthesizing genome becomes a post-replication genome, so this will be the green arrow. And when a cell division occurs, a post-replication genome becomes a template for the next generation. And all of these types can produce synthesizing genome. So synthesizing genome can start replication again and produce other synthesizing genome. So this is our initial model. Does it make sense? Again, please interrupt me if there are questions. Okay, so again, these are the equations implied by this simple scheme. So again, it's a simple linear rate model. And if I solve this model, I will find that there will be an exponentially growing phase in which all the genome types are growing exponentially at a rate k. So n t of t goes like e to the kt, and similarly for all of them. And the relative fraction of these three genome types are constant. So there will be a steady growth regime. So this means that this rate of production of synthesizing genomes corresponds to the growth rate of the population in this model.
Can I ask a question?
Yes, please.
Yeah. So what is the difference between this template and the post-replication? I see that both can generate, both can synthesize.
Yeah, concretely they are the same, so they are complete as well. But I think one way of seeing is that this is a way of keeping track of the number of cells. So the number of templates corresponds to the number of cells. So the idea is that each cells must be given a template. They must have one complete genome, otherwise it cannot function. And then it can create other complete genomes, but these are considered as post-replication until a cell division occurs, and then these are passed.
But both template and the post-replication can synthesize gene.
Yes.
Okay.
Yes. We can generalize the model assuming that the rate at which these three types of genome produce new synthesizing genome is different, but that complicates the math and it doesn't change the basic physics. So we'll just keep it simple like this. Okay, so now I come to the question that I received before, which is we want to zoom in these synthesizing genomes and look at their size distribution. And in particular, we want to look at their dynamics. So what we say is that these two replisomes proceed at a certain speed that in principle can depend on their position that we measure as the distance they have traveled from the origin. And they are also subject to, there will be a 1D diffusion constant. So their velocity is subject to some stochastic fluctuation. So in other words, there will be two Langevin equations governing the dynamics of these two genomes with a space-dependent velocity, just to keep it general, and a diffusion term. Right? And now I'll tell you how to somehow match this dynamical model of the replisome with this population level model of the genome population. That's the most interesting part of the model.
But Simone,
yes.
So this X1 and X2 at the end of the process or somewhere, like they have to meet each other, right?
Yes.
So will there be any constraint regarding?
There is an absorbing state at some point. I will show you it in a couple of slides. I hope it will be clear. So let me put that question aside. I will get there. Thanks. That's a good point. Okay, so again, let me discuss boundary conditions in a minute. But first, what I want to say is that there will be then a density of incomplete genomes. So this is the number of incomplete genomes at a time t, whose first replisome has copied the distance x1 and the second replisome has copied the distance x2. And this is such that the integral, its integral is equal to the total number of synthesizing genome that I introduced before. And you can see that in the extremes of this integral, I impose this condition that the sum of x1 plus x2 must be equal to L. Okay. Yes. So these two Langevin dynamics essentially imply a sort of Fokker-Planck equation for this density, because the coordinates evolve with the stochastic dynamics, and so I have this this equation for the density of incomplete genomes. But I'm saying a sort of Fokker-Planck dynamics because, as I said, this is not a normalized distribution, because the integral of this function is increasing exponentially. It's not a constant. So I have the total number, as the population is growing exponentially, the total number of incomplete genomes is growing exponentially as well, and therefore also its density is growing exponential. exponentially. So what I will do is to define a normalized density, which is this non-normalized density divided the total number. So P is normalized to 1 now. And I use the fact that the number of incomplete genomes is scales like the exponential of KT. And if I plug this equation inside this relation, what I obtain is an equation for P, where I have the same kind of Fokker-Planck dynamics minus a dilution term. So this dilution term comes from the fact that the total number of genomes is growing exponentially. I mean, this is just a simple calculation. You can just plug this relation here and use this fact.
So this K is the same K, right? As
Yes.
your okay.
Yeah. So here it comes. So this is again the answer to the previous question. So we have this equation, which is the central dynamics that we want to study. So again, this is it's a Fokker-Planck equation with this dilution term, but we can interpret this dilution term as something described as in a stochastic resetting. So if I want to think of this as an equation describing a stochastic process, there will be I would have to reset the trajectories to the origin with the rate k. So let me represent this process in what I would like to call the Replisome phase space. So this is the distance traveled by the two Replisomes. And the process terminates when both Replisomes have copied the entire genome. So x1 plus x2 equal L, which is this red line that is treated as an absorbing state. So if I didn't have this term, I would just have a normal drift diffusion dynamics in this space. But because of this term, I have superimposed a resetting term. So the process starts from the origin, and it can be reset to the origin either if it reaches the absorbing state or with rate k. So this would be this blue trajectory here. And the stationary distribution of this drift diffusion resetting process is the steady distribution of replisome of incomplete genome length, the replisome positions, and therefore incomplete genome size that I should expect in my exponentially growing population. So that's the analogy with stochastic resetting.
But this equation, like the total probability is not conserved, right? It decreases with time.
So the other thing that I didn't write here, this is a good point. So there is an influx in the origin. Okay. So there is a source term, because you will get this firing creation of new replisomes, new duplications. So there is a source term which has a complicated expression, but essentially we match it by imposing normalization. So P has to be normalized because we showed it in the previous slide. The P is normalized. So essentially, normalization tells me the magnitude of the influx term. Thanks for the question. This is a good point.
So with this influx and with this decay term, that minus Kp, so in this this system will reach a steady state where P, the probability is constant over time.
Exactly. So this is the dynamics. Okay. So I mean, this is true for any kind of Langevin dynamics. I still have to choose this function V and the diffusion coefficient. So the last thing is that once I have this stationary distribution, I can use it to predict the DNA abundance distribution. So the idea is that if I had to measure the abundance of DNA along the genome, I will have that the templates and the post-replication genomes will give me a uniform contribution. And then I will have this peak that is created by the replicating genomes depending on their size. So this can be formalized by this expression where the abundance of a certain position I includes a constant term, which is the probability of picking a complete genome. And then I had to include the probability that that position I has been copied by the first replisome and by the second replisome. And this just involves essentially integrals of this steady distribution that I computed from my stochastic resetting problem. So essentially, yeah, we get that this distribution is related with the integral of the steady state distribution. So that's my theory. And the nice thing is that the experiments that I showed you is from 2014. Sequencing techniques have been improved, and there are lots of things that we can do to have more precise curves. So these are actually experiments that we performed here at OIST by growing E. coli at different temperatures and using a number of tricks, for example, these PCR-free techniques to essentially reduce the measuring noise. So you can see that we can obtain now very precise measurements of the DNA abundance distributions, and we can clearly see that there is a difference when measuring this distribution for colonies growing at different temperatures. So now we have all the tools that we need to try to do inference. So I can try to make guess of this function v of x, this function d, solve the stochastic resetting problem. From the solution, compute the DNA abundance distribution and compare it with experiments and test the validity of my assumptions. So this is what we are going to do. So the first thing we can do is to say, let's assume that replisomes are perfect machines that travel at a constant speed with zero diffusion coefficient. So if you do that, then the stochastic resetting equation becomes a very simple equation. In particular, you can show that it factorizes. You can compute the steady solution that is simply exponential, and the DNA abundance distribution is exponential as well. So this would be the simplest case. If I try to fit it, this is pretty good. So you can see already that the results are pretty compatible with the idea that replisomes go at a constant speed. We can do this fit for different temperatures, and we can find that we can measure the velocity because the growth rate can be measured independently during the experiment by looking at how the colony grows. And so we find that the velocity has a Arrhenius dependence on the temperature. And this is interesting because this Arrhenius dependence is different from that of the growth rate. So if you see an Arrhenius plot of the velocities in logarithmic scale versus 1 over t, this is the growth rate and this is the velocity. So these are two different slopes. And this is important for biology because essentially this is the reason why you have to do this parallel replication, because somehow when the growth rate increases, the velocity of replisome does not increase at the same pace. So these two curves are not parallel. So it cannot catch up with the speed of replication when the temperature grows. So something I want you to notice is that if you look at this fit, this is good, but if you squint, you will see that here the points are slightly below the curve, and here are slightly above the curve, and here are slightly below. It looks like a small effect. So here is the same, slightly below, slightly above. But the effect is there, and actually the curves are rather precise. So I want to show it this effect clearly by dividing the experimental data from the fitted over the fitted theory. And these are the results. So now I'm showing you the data divided the theoretical curve. And you can see that the center is the origin. And so you see on both sides the portions of genome that are copied by the two replisomes. And you can see there is this oscillatory behavior, especially at high temperature. It almost disappears at low temperature. And this is highly repeatable. So we repeated the experiments at each temperature and we saw always the same pattern. So we try to understand what this is, and what we came out is, let's say that the velocity is not constant, but there is an oscillatory term. So it oscillates along the genome. And in this case, we also consider the effect of diffusion. So we added the diffusion term. So we couldn't solve the equation corresponding to this choice exactly, like in the constant velocity case. And therefore, we did numerical simulations exploiting this analogy with stochastic resetting. And what we found is that this model fits the data much better. So this I can show you in the bottom row fits of the experimental data at different temperatures, and on the top row using the ratio with the constant velocity model to appreciate these oscillations as before. And we should say that, of course, here it fits better because I introduced 4 extra parameters, this delta, which is the amplitude of the oscillations, the wave number, the phase, and the diffusion constant. But the Archaic information criterion supports that these extra parameters are justified, at least these 3 extra parameters. I will talk about diffusion later. So this model, the quality of the data somehow justifies this description over the simple description with the constant velocity.
Simone?
Yes?
So these oscillations mean that Replisome is going back and forth along the genome to
No, sorry, it's going always, it cannot go back. It goes in the same direction. It means that it speeds up and then slows down. It speeds up and then slows down.
Okay, so it is always some positive velocity, but
it's always positive.
And then
yeah.
Okay.
In fact, here it is. So this is a so since the velocities are quite different at different temperatures, I show you the velocity as a function of the position divided the average velocity at a different temperature. And you can see that these oscillations are the order of, say, 20%. And we were wondering, so what is this? Like, this is a kind of strange result. This is not really what we expected. So because this is a very long wavelength oscillation. So what causes the replisome to speed up for hundreds of thousands of base pairs and then slow down again? But we found a result that it's quite fascinating. So there was a previous paper a few years ago which measured the mutation rate along the genome on a mutant of E. coli, which has a damage in one of the mismatch repair mechanisms. So essentially is a high mutation rate strain. And what this study found is that there is a wave-like pattern of the mutation rate along the genome as well. So I show you this green dot, which are the results of this other study using a different strain of E. coli. And the top is the result of our inference. So you can see that this wave pattern in the mutation rate is very similar to the pattern we found. And this is quite suggestive because theoretical studies, like the studies I was mentioning in my introduction, suggest that there should be a trade-off between speed and accuracy. So in other words, when the replisomes go faster, they are expected to make more mistakes and therefore generate a higher mutation rate. So this is consistent to what you see in this figure. So this suggests that also in these studies, there might be a higher velocity in this region, and therefore this might cause these periodic changes in the mutation rate. So this is a pretty nice support that this is a real thing, it's not an artifact of our model.
So this oscillation is related to proofreading. You are mentioning like that, like is it?
No, the picture that emerges is that for some reason the speed oscillates, and it causes variation in accuracy as well. This is what I'm saying. Okay. So there must be a dynamical reason why there are these oscillations, and as a consequence, the accuracy oscillates as well.
So you have 3 minutes plus 5 minutes for questions.
Okay, so let me wrap up. I'll just tell you a few more things about these results. So one is briefly about the causes of this oscillation. So we don't have a definitive answer, but our suggestion is that since these are occur at high temperature, and at high temperature we know that replication occurs in parallel, this might be caused by competition among replisomes. So there might be a situation in which when the next replisome starts, it takes away resources from existing replisomes and therefore causes them to slow down. So they will be competing, for example, for the same nucleotides. But this is just an assumption of this model. It supports, it's consistent with our results, but we don't have a definitive answer to this question yet. I'll tell you a few things about our fit of the diffusion constant. So we find that the diffusion constant is quite small. It's on the order of 1 kilobase per squared per second. In some cases, we can't even fit it. So for some realization, we find that the Akaika information criterion tells us that the diffusion coefficient is practically 0. It's also on the order of the equilibrium estimate if you use the Einstein-Stokes relation, which is remarkable given the fact that these machines are traveling so fast. And coming back to my original question, this leads to estimate an uncertainty on the meeting point of the replisomes, which, by dimensional analysis is this quantity of the order of 10 to the 5 base pairs, which is a quite small number. I'll tell you briefly about a failed attempt. So we're thinking about, since now we know the average velocity and the diffusion coefficient of these machines, we were thinking about thermodynamic uncertainty relations. So whether somehow the ratio between the diffusion coefficient and the velocity squared has something to do with the dissipation of these machines. Again, this is, I think, an interesting question since this is a workshop on stochastic thermodynamics. But if you plug in our typical numbers, say at a high temperature, the velocity is order 10 to the 3 base pair per second, and this is the diffusion coefficient, then you would say that the minimum dissipation per monomer required to achieve this precision is order 10 to the minus 3 kb, which is way, way smaller than how much these machines consume. So these machines consume several NTPs per step, and each one would be 25 kb. So this problem seems to be very far from the bound set by thermodynamic uncertainty relations. And it'd be interesting to see whether one can formulate some different kinds of bounds inspired to thermodynamic uncertainty relations, which is more tight for this kind of biological question. I conclude. So in this presentation, I introduced a method to study the dynamics of bacterial replisomes, the detailed stochastic dynamics that they undergo in vivo. We found this dependence of the average replisome speed on temperature, and the most surprising fact that we found is these regular speed oscillations around the genome that are correlated with the mutation rate. So the work, as I was saying, is under review. And one thing we would like to do in the future is to adapt these formulae to understand mutants, which have a very different shape of the DNA abundance distribution. So I would like to thank my group at OIST for the support. So this was mostly work by Deepak Bhatt, that is now assistant professor at the Law Institute, and my funding sources, and thanks for your attention.
Thank you very much. A very interesting talk. So I guess we can take few questions.
Hello, Simone.
Yes.
So you have the equation of motion for this, right? This dx1 dt. Then there is a noise term on the right hand side. So can you just elaborate it? What is the noise here? Like, is it only thermal noise or some other possibilities, or it's only thermal? That Xi term in this replisome dynamics?
That's a very good question. So I think of it as a thermal noise, but this is a very large machine, and there might be, so normally if you, in some cases, replisome dynamics is very difficult to study in vivo, but for example, if you look at trajectories of some of these machines, they might have other sources of fluctuations. For example, they can stall. So sometimes they just get stuck for some reason, and then they continue again. So we don't have single molecule tracks. We are looking more of an ensemble view. So it might be that somehow this diffusion term, you should think of it as not only as a thermal diffusion, but something somehow effectively incorporating all sources of fluctuations that these machines suffer in a way. Yeah, but this is a good question. I mean, at a basic level, you can think of it as just thermal fluctuation, but in principle, you can absorb other effects inside it.
So you mentioned in the beginning that like before one completion of one synthesis, there are multiple syntheses happen. So can one think of it like multiple random walks at the same time? I mean, can it be an extension or, I mean, independent random walks?
In a way, you can think of this as I mean, it's a very biased random walk because the velocity is much larger than the diffusion coefficient and the number is varying. So you can think of this as a exponentially growing number of very biased random walks, two-dimensional random walks. Yeah, that's one way of seeing it. Yeah.
Okay, so it's like a branching random walks.
So what's branching is not the random walk itself; it's the number.
Yeah,
of random
yeah, branching plus
yeah,
random walk.
sense, yes.
Yeah. Okay.
With accretion rate, yes.
Oh, yeah. Okay. I think there are.
Simone,
yeah, you have a question, Sourabh?
Yeah, so this velocity oscillations that you showed, that was for any one of these replisomes.
Yeah, it's quite symmetric. If you see, for example, here. So here in these plots, the origin is in the center, and it's like a mirror. So left is the first replisome, and right is the second replisome. So it looks like they are subject to the same dynamics. There's no, we couldn't see any difference between them. I mean, it's quite a symmetric pattern.
So Edgar, if you have a quick question, we can take it. Otherwise,
very quick. So,
yeah.
So what makes you discard a possible underdamped Langevin model? Because you are using overdamp, so for the velocity, but. So do you have the autocorrelation function and it motivates you to use overdamp, or?
I would guess that scale things tend to be underdamped. I'm not sure if inertia is important, but I must say I didn't really make like an estimate of. Do you think that there could be?
I'm not sure. I was guessing that you have the autocorrelation function and you concluded that.
No, I mean, it's mostly for simplicity and thinking that this is a molecule, so it's mostly underdamped. But I didn't really think whether underdamped model would be appropriate.
Okay.
So yeah, okay. Let's thank our speaker again, and then
Thanks a lot.
thank you.